Hello, everyone. Welcome to Room Now's coverage of ACR 2025. In this session, we'll be reviewing abstracts and presentations on CAR T cell therapy and cellular depletion therapies. We've got a faculty who were out there scouring the abstracts and presentations for insights and what was notable. We're going to review their favorites in this podcast and videocast. I'm Jack Cush with Room Now. I'll ask our faculty to introduce themselves. Akil?
Yeah, thank you, Jack. I'm Akil Sood. I'm a rheumatologist at Washington University in St. Louis with an interest in lupus.
Okay. Yus?
Hello, everyone. My name is Yusuf. I'm a rheumatologist in Leeds.
Yes. Philip?
Hi, I'm Philip Mees. I'm a rheumatologist based in Seattle, Washington, and director of rheumatology research at Providence Swedish Medical Center.
And Al?
Hi, I'm Al Kim, adult rheumatologist, also at Washington University School of Medicine, also interested in lupus, where I direct the Lupus Center.
Excellent. All right. So we're going to do a few rounds of discussion and presentation. Let's begin with Dr. Sood.
Yeah, so I think during ACR25, there are definitely a lot of abstracts on CAR T, and I think we definitely seen since the landmark case series out of Erlangen in 2024, we've seen an explosion in new CAR T therapies since then. And I think the Erlangen study was there was a really robust study showing efficacy and safety. And abstract 0663 took a different approach. It took an off-the-shelf approach with CAR T. So essentially what it was is compared to autologous CAR T, extracting individuals' own T cells, engineering them to target the CD19 T cells. They're taking an off-the-shelf approach, meaning they're extracting T cells from a bank of induced pluripotent stem cells and engineering them to target the CD19 B cells. And the advantage of this is that the CAR T is accessible and available. And for the patients, this is wonderful in that they have a much shorter hospitalization for monitoring. And so this abstract 0663 evaluated the safety and efficacy of off-the-shelf CAR T in patients with autoimmune diseases. And to date, they had patients with lupus that were enrolled, and their inclusion criteria were those that had a high disease activity based on SLEDA and BILAG scores. And the patients were allocated to two regimens. One regimen involved a limited lymphodepletion, meaning they didn't receive fludarabine, but they received cyclophosphamide only. And then the other group, which was two patients, had no lymphodepletion at all. And to date, they've had 10 patients enrolled in this ongoing phase 1 trial. And they found that in terms of efficacy, at the 6-month mark, all the patients had improvements in their CLAD2K score at 6 months. But after that is when we kind of start to see things kind of vary. At the 9-month mark, one patient actually had a spike or a rebound in their SLEDA2K at the 9-month follow-up. And then when it came to patient-reported outcomes, all the patients had improvements in their fatigue, especially those that received the limited lymphodepletion. The patient who didn't receive the lymphodepletion had a improvement in fatigue based on passive fatigue at the 3-month mark. So a little bit shorter time. And then when it came to the B cell depletion, it's kind of where we saw differences in terms of B cell repopulation, both for those with limited lymphodepletion versus no lymphodepletion. Those with limited lymphodepletion had a much longer time, up to 3 months of repopulation of their CD19 B cells. But on the other hand, patients who did not receive lymphodepletion had a much shorter time of repopulation of these CD19 B cells, as much as just a few days post-infusion. And so what I thought was just really interesting was that this off-the-shelf is a really exciting approach. It seems it could be a more patient-centric approach, meaning it's readily available, accessible, and the T cells and even much shorter time for hospitalization for our patients. But I think there's still a lot of questions that need to be answered, especially longer-term follow-up, looking at other autoimmune diseases, and even the question of lymphodepletion that they did tap into in this abstract.
So there's here a number of different approaches to cellular depletion, and we're looking for the advantages and disadvantages. Anyone, others want to comment here on what the advantages and disadvantages of this off-the-shelf approach is? Use?
Yeah, so this is really interesting because it does help that phase whether we can, give therapy without doing lymphodepletion. And also the interesting thing is, you can get it off the shelf from this pluripotent cell. I think we still need longer term data about this, whether, in the long run, whether there is a problem with the graft versus host disease or anything like that. Certainly from the preliminary data that they, reported, they couldn't see one. But it is really interesting. It's like you're trying to go back, from your ANA test, isn't it? With your Henry letter like cell come from one person and then you then multiply it many, many times. So I think this is what happened. They're trying to do in this, iPSC pluripotent CAR T. So yeah. Mhm.
Philip, you've done tons of trials. At this stage of the game, when we have such a limitation on numbers of patients, what are we learning?
I think that the autologous CAR T programs are giving us the gold standard of what to expect. We certainly saw that in the results from the Erlangen studies. I think that, as mentioned, the allogeneic approach is going to be more practical and feasible. You don't necessarily have to be in a major center and automatically partnered with your hemonc colleagues with this off-the-shelf approach. But what we're missing is what's the right dose and how durable is it going to be in comparison to what I call the more gold standard autologous approach.
Yeah. Dr. Kim, any comments?
Yeah, so a couple of them. So I think you brings up an interesting point about the LDC issue, lymphodepleting chemo. That one patient that they had in that FATE study that's been described, didn't do so as well as everyone else. It's stuck in LLDAS. The CAR T cells didn't expand very well. The B cells didn't really reconstitute the way it did with people who got LDC. But generally speaking, I'm very high on allo, largely because there's going to be a cost savings, substantial cost savings associated with this, because there's no individual drug manufacturing. Most of the costs come through hospitalization, which probably hopefully will drop.
Yeah, I like that as well. All right, Dr. Yusuf, why don't you go next?
So talking about deep tissue depletion, so I just want to bring an abstract 26995. This abstract was awarded as the Emerging Investigator Excellence Award, and it was actually presented at the very last session of the ACR, where a lot of people have actually flown home. So I found it really interesting. So basically, this study is again from Professor Schatz's group. So they tried to look at the depth of tissue depletion in a few treatments. So basically, they did a biopsy, at least 3 to 4 patients per group on the lymph nodes, for people who received treatment, including rituximab, which is your old anti-CD20 monoclonal antibody, and you have the newer humanized type 2 anti-CD20 monoclonal antibody obinutuzumab. They also compared blinatumomab, which was your bispecific T-cell engagers monoclonal antibodies, and also patients who received the CD19 autologous CAR T-cell. So when they look into the lymph nodes depletion, what they found that only patients treated with the CD19 CAR T cell consistently achieve deep tissue depletion. All other protein-based depleters reduce, but not complete as CD19 CAR T cell. And of all these protein-based depleters, so the one that comes sort of slightly closer to CD19 CAR T cell was obinutuzumab. So yeah, so I think I found it really interesting that not just the blood, actually we do have some evidence that all this treatment actually leads to deep tissue depletion, which is really an important key therapeutic target here. Yeah.
So was this done for just a comparative analysis, or was it done with the idea that maybe we would be using these therapies together or in place of one another?
No, I think it's more the mechanistic study just to demonstrate that, a lot of study before just showing blood depletion, but then people say did you also deplete in the tertiary lymphoid tissues? And that's what they wanted to show here. Yeah. Mhm.
Excellent. Any other comment?
I think I haven't used a slide that shows this degree of depletion, and exactly that. It's a comparative way of looking at the total effect, and I think it visually it's a great teaching slide.
Excellent. All right, Philip, what do you have?
So I really appreciated a review talk that was presented by Joan Merrill in a session called The New Rules of the Road, an update on CAR T mediated immune reset for SLE, that was moderated by Sarah Sheik on Sunday at 1 o'clock. And Joan started by presenting a just published this month systematic review of CAR T results in lupus. There were a total of 16 studies, 13 targeting CD19 and 3 CD19 and BCMA. There were 12 of these that were autologous and 4 that were allogeneic. The total number of subjects was 145 patients with lupus. Their baseline SLEDAI score was 13, and then after a 6-month period, this decreased to 2.3 on average, and after 12 months, decreased to 1.4. 70% of the patients achieved DORIS remission. LLDAS was achieved by 89%, and drug-free remission by 84%. These numbers correspond to what we're experiencing in our autologous as well as allogeneic trials that we're doing. So it was interesting and reassuring to see that there's a consistent degree of result across multiple studies that are occurring. There were side effects that were detailed, and CRS, or cytokine release syndrome, was seen in 56% of the subjects. 98% of these were grade 1 or 2. And when I'm working with my hem-onc colleagues who are more used to more severe CRS or possibly ICANS with their lymphoma or myeloma patients, they are impressed by the fact that we're seeing much less of this kind of side effect in our lupus population, as well as scleroderma and myositis patients. And presumably this is partly related to the lesser amount of B cell volume that's present in these patients. There were 4 cases of ICANS, the neurologic toxicity, 1 each in grade 1, 2, 3, and 4. And when it gets up to 3 and 4 with this particular side effect, this is an important issue to really focus on. 7.6% of the patients developed serious infections, and there was 1 case of fatal pneumococcal meningitis. So in summary, on the one hand, we saw terrific efficacy that came close to what we saw in the original Erlangen studies, but there's also this set of side effects that we're seeing.
Al, what do you think?
Yeah, so this is such an important review that Akanese's group has done. I think it does a couple things. First, it actually helps restore some faith in what we should see globally with these trials. As people have mentioned before, Georg's Erlangen study, the first one, the council study, where 10 out of 10 hit DORIS, I'm not sure that is actually realistic, and I think there are reasons for that. The rest of the experiences that people are having, including ourselves, Phil's, others, are more in line with what this review is and helps set expectations.
So what would then be, for the others, what would then be the side effect you're most concerned about going forward? We're not seeing yet what the FDA was worried about with all the oncology CAR Ts with now black box warning for secondary malignancies and whatnot. So that's good news, but still we're early in the process. What is the concerns that you as a group have, you've been studying this. What would you want to be most concerned about or worried about going forward? Akil? Oh, you're muted. All right, we'll come back to you. Use.
Yeah, so I think well for me I still want like a longer follow-up data, even particularly in terms of efficacy, because really and what the CAR T cell principle trying to sell is this profound depletion blood and tissues, and hopefully after that be able to potentially immune reset patient to be remission free on drugs. However, we have seen, a few, although it's still smaller number, cases of relapse, after these therapies. So really, at, what costs, we are putting our patients in to go through all this, potentially, toxic medications, and then at the other side, in a few years down the line, they will relapse and need potentially further treatments. Whereas we also now have like a better monoclonal antibodies, for example, the obinutuzumab that can do almost as good. Yeah, so I think that's quite a question from my point of view. Yeah.
Philip, not a lot of mention of serious infections and opportunistic infections. What was said about that in your session?
That they can occur and need to be monitored for. And when a patient presents with fever, is it CRS or is it a brewing infection? So these patients need to be monitored and looking for evidence of infection, partly because of the cytopenia and hypogammaglobulinemia that can sometimes occur. Just yesterday, I had one of our patients, we needed to give IVIG because of low immunoglobulins. I think it's so these are real things. We're going to need in-services from our HEMONC colleagues to really get up to speed with how to look out for these and monitor for them. It's way too early, I would say, before we can tell whether they're going to be CAR-associated malignancies. And so that's why we're being asked to monitor patients for a total of 15 years after the
Oh
treatment.
my. Okay. Dr. Kim, what do you have?
All right, so a lot of the discussions in past ACRs and EULARs have been focusing on lupus with increasing myopathies, scleroderma, and whatnot. But now we finally have some RA data that's out there. So it was abstract 471 looking at Kyverna's autologous CD19 product against in RA. So this is going to be ACPA positive, as one should imagine in these type of trials, and difficult to treat RA. So phase 1 study, six people were enrolled. Primary outcome is going to be safety, obviously. They all received lymphodepleting chemo. So not surprisingly, 6 out of 6 people got transient cytopenia. Fine. 6 out of 6 people, though, did develop cytokine release syndrome, which is going to be higher than what Philip just talked about in terms of that meta-analysis. 4 were grade 1, 2 were grade 2, but so this is a little bit higher than we expected. Now, in terms of efficacy and PD type stuff, so interestingly, most people, almost all of them, drop their ACPAs to either very low or undetectable levels, all 5 out of 6. But only 4 out of the 5 hit ACR20. Right now, granted, 3 out of those 6 are haven't hit the 16-week level time point yet, the 4-month. So maybe it's a little too early. But the other 3, there's one that did not hit ACR20, and I believe they told me it was largely because of pain. It opens up a whole bunch of different questions. For me, first of all, is this the right target? Should we be targeting something that's more antibody secreting, although their ACPAs do drop a lot, so maybe 19 might be sufficient. B, are ACPAs pathogenic in these type of diseases, right? Maybe in terms of the inducing disease, but its removal once the disease has started, is that sufficient, right? So I think that's the other beauty about these studies, right? Like we think very simplistic about how these model out, but the nuances that we're seeing in some of these very small studies are intriguing.
Yeah, I'm thinking as you're talking about this, clearly there's a biologic effect. Clearly, the issue is, does that correlate with the clinical outcome you desire?
Mhm.
And we don't know about that lowering of seropositivity, but there was an abstract by Smolin's group 2 years ago about drugs that lower rheumatoid factor have better outcomes, better responses. I don't, not sure I agree with that. but they have the data and I have an opinion. So I'm waiting for that paper to come out. But nonetheless, if we can do that here, maybe we can. So tying together biologic responses and clinical responses for whatever disease is going to be important going forward. All right, let's go one more time around the horn with Akil.
Yeah, can you hear me?
Yes.
Oh, perfect. Yeah. So the other abstract was abstract 2465. This was looking at CD19 BCMA dual targeting CAR T in patients with refractory lupus. This was a single center investigator initiated trial. So essentially, they had 10 patients with refractory lupus, SLEDAI score of SLEDAI score, 2K score ranging from 8 to 20. And in this one, all the patients received lymphodepletion, meaning cyclophosphamide plus fludarabine. And this was followed by a single dose of a rapidly engineered autologous CAR T, the CD19 BCMA CAR T, and was given at 3 dose levels. Over the primary outcome was looking about the tolerability and the safety of CAR T, but they also evaluated the efficacy based on the SRI-4, DORIS criteria, and renal response. And they found that by week 24, all 10 patients achieved their SRI-4 response by week 24, and then 5 of the 10 patients met DORIS remission criteria by week 24, 6 by week 36, and it continued till week 48. Or sorry, week 48. And then in terms of the disease control remission, one patient did flare at week 40, and even the patients with they found that 4 of the patients actually experienced proteinuria. And with that proteinuria, there was a concern is that if there's a relapse of their lupus, but when they did a renal biopsy, they actually showed that there was resolution of acute inflammation and there was a decrease in immune complex deposition. So I thought was really interesting. So overall, I thought, and also one thing was that the safety profile, it was relatively safe. 6 patients had grade 1 cytokine release syndrome. 1 patient had grade 2. No reports of ICANS or higher grade CRS reported, with common side effects as previously alluded, including cytopenias and hypogammaglobulinemia. And so while overall these findings are exciting, I still think we still need ongoing enrollment as well as longer term follow-up with this trial.
So albeit small, 10 patient cohort, no infections, no major issues on immunoglobulins and, that you would worry about because BCMA is going to go after plasma cells and is clearly going to affect immunoglobulins and antibody production. And the question is that going to be great or disastrous? So, and there are a few of these CD19 BCMAs that are in play. So we maybe need to pool this data to know how safe this approach is. Any other comments on this?
Well, I think there's somewhere in between, Jack, where yes, BCMA by ablating plasma cells, you're likely to lose your viral memory for mumps, measles, etc. But we can monitor that, and we can also revaccinate. So this is something that we're building into the next program we're going to be working with, where we do a dual CD19 and BCMA, where we absolutely caution the patients that we're going to need to revaccinate.
I like that approach. Al?
Yeah, so it's interesting because I think we're starting to see some diversity in outcomes with BCMA targeting. If you look at the engagers with Teclistamab, that is almost universally going to drive hypogammaglobulinemia, and there are some drops in protective antibody titers that was presented at ACR. So it's CAR T versus engagers for BCMA is a really interesting question because you can revaccinate it, but in engagers, you're probably also gonna have to redose. So where does that leave you in terms of protective immunity? I'm not sure, right? And of course, all of these were generated for multiple myeloma. Is this over engineering for autoimmune disease?
Yeah, good point. All right, Dr. Yusuf.
Yeah, can I add that one as well? So, based on this abstract, I did manage to have a discussion with the presenters. I mean, they were really busy with a lot of people coming to their stands. So I was particularly concerned, regarding to the risk of infection and also hypergammaglobulinemia. They did mention that they did see a few, although they didn't put that in the poster. And also, and I think a few patients also had treatment with IVIG as well. So I think we're trying to hit patients as hard as we can, but really, at what cost? And we have to find the balance between infection profile and also, deep depletion to remove all the antibodies as well. So.
Okay, Hughes, what's your next one? A short one.
Yeah, so I'll just trying to move. We talked about the cytokine release syndrome earlier. So this is not CAR T. This is one of T cell engagers. That this is like a something that we can look forward to in the future. So I usually don't really want to discuss things that are not in human studies yet, but I think this is something that I found it super sexy and super cool. So the abstract is 1535. So for this T cell engagers, the bispecific T cell engagers, so instead of engaging to the CD3, what they did is they engaged to pan gamma delta T cell receptor. So this is really super sexy. So they did a comparison using in vitro blood from patient treated with this compound, also compared with other BiTE and mono antibodies available, such as the blinatumomab and also mosunetuzumab. What they found that the degree of the B cell depletion achieved in the bloods were comparable between these three. However, from the safety profile, from the cytokine release syndrome in a perspective, they found that no issue with this pan gamma delta, because you don't see so the interleukins 6, 10, interferon gamma all completely reduced from this from this blockade. So I found that this really interesting. They're now progressing this to potentially phase 1 in the next few months. So something that that we can watch out this mechanism of action. Yeah.
Can we realistically expect there would be less or no cytokine release syndrome with T cell engagers or BiTEs?
Yeah, so from their in vitro data, they showed like absolutely just the level was absolutely suppressed, from the engaging. So yeah.
If that were true, that would be great. That's why we do the research. Okay, Philip.
So one of the side effect issues that has been emerging recently, highlighted by Melanie Hagen from the Erlangen group, is something called LICATS. And it stands for Local Immune Effector Cell Associated Toxicity Syndrome. Say that rapidly. So this is a fascinating phenomenon, and we have seen it in at least 2 of our patients, where about 10 days after cell therapy on average, you see a flare of arthritis, skin rash, kidney symptoms and signs like proteinuria. And at first, when I saw this occur in our first patient, about that time, I thought, well, this must be their underlying disease flaring because of the washout of their previous medications and before cell therapy had fully kicked in. But Melanie has highlighted the point that the conditioning regimen is probably still having some effect to suppress the actual disease itself, the lupus. So it's probably not a flare of lupus. And so she has hypothesized that this represents a reaction to the dead B cells in the field of battle. So in the places where the patient had arthritis, where the patient had skin rash previously, where the patient had kidney involvement. And it was not associated with drops in complement or rising DNA antibody. And it was transient, just lasted on average about 11 days, and was easily treated with steroids and or tocilizumab. So an interesting problem to be aware of and look for, but very manageable.
So how can you know that it's related to B cell death? What's the biomarker or the indication there?
So a couple of the points that I just made, i.e. that they're unlike the patient's previous low complement levels or high DNA antibody associated with their manifestations of disease. In this case, it was not. And the fact that it was transient, easily treated. What I thought you were going to ask is, how do you differentiate this from some other issue arising, including differentiating it from infection or differentiating it from a flare of the disease. And I think we just have to be aware of it. And if it persists and is associated with rising antibodies or low complements, then we have to think it could be flare of disease.
No, my question though had to do with the fact that all these therapies are going to deplete, and then you assume depletion is associated with cell death. So why is it that what is it biologically that's different about these cells that leads to the syndrome?
I don't know the answer to that, Jack.
But I'll make something up.
Yeah, I'll make something up. One could hypothesize that tissue burden of B cells could be an answer, right? We don't know that answer yet, right? That would be my hypothesis, though.
Yeah, interesting.
So she said this was seen in 77% of their 39 patients. So it's not an unlikely phenomenon.
Okay. All right, I'll wrap things up.
Can I add that one?
Yes, please.
Yeah, so I think this is really interesting, and it just brought me back to to the old story of rituximab that when I first started using it when I first started doing my PhD a few years ago. So my groups Professor Ed Vital and myself, like when we used rituximab before, we did found that patient had this skin flare shortly after treatment with rituximab, and we did do some skin biopsy, which did show some active lupus, and some actually changed phenotype from acute cutaneous lupus before when we first started treating it, and they changed into subacute acute cutaneous lupus, and also some patient changed into CCLE as well, chronic cutaneous lupus. So that was quite interesting, but that indicates sort of the disease flare, post B cell lysis. Where also in this group, I did have a chat with the investigator as well. They said they did few skin biopsy, and they said the biopsy wasn't abnormal in terms of active lesions. So yeah, so that's just an interesting phenomenon, to be described as one.
And by knocking out Bs, your effect on Ts is going to be variable. And, how do we know this is not just a rebound effect of T cell compensatory activities and what that does to the disease? So it's a complex story that needs to be, I think, but I like that you've given it a name and a hypothesis so people can work on this. All right, Al.
All right, let's wrap this up. So, the older rheumatologists may remember when Jonathan Edwards first proposed this concept of restoring or resetting B cell autoimmunity with B cell depletion. And obviously, of course, rituximab was the thing at the time. And that never got fully realized, maybe because it doesn't deplete well in lymph nodes, where a lot of the immune responses start. So abstract 2442 comes from Erlangen. This is the largest study that I'm aware of and the longest study looking at B cell subsets only in the blood, but they did some really great work with spectral flow cytometry and single cell RNA-seq. Bottom line, for patients with lupus, they have really skewed B cells. They're very heavily double negative 2 memory B cell phenotypes, and they become plasmablasts that are 95% of them are autoreactive. And then when you look at B cell receptors, that the autoreactive B cell receptors dominate the scene. In healthy people, it's complete opposite. Naive, no autoreactivity. The bottom line, 12 months after getting CAR T cells, they look like controls. And interestingly, of the B cells that do come back and they're activated, they're all activated through pathways that are not recognized to be autoimmune, i.e. extrafollicular pathways, but rather the traditional ways of being activated, germinal centers. So there's this durability in B cell biology in this quote unquote resetting that could explain the durability of clinical response.
So that data is developed on the Schett Erlangen group?
Yeah, so this is multiple trials though, not just Castle, but also other ones.
Okay. Well, that's great. And what I like about this, and what I hope is going to happen in this field, as an outsider watching, is that there'll be a lot of cross-talking, a lot of cross-learning, because everybody's experience is going to be small. And right now, there's 40 plus, agents that are in play by, actually, it's much probably larger than that. And everyone's got an N of, single digits. What is the largest trial and how far out are we going? Al, you following this, do you know?
The answer is yes, I don't know.
Good answer. But we are getting out there with numbers, in a few cases, but when we have high numbers, we have no controls. And so I definitely think this is a big step forward for the field and what you guys have covered and reviewed here today. And obviously next year is going to be a real eye opener if this year wasn't. I want to thank everyone for their efforts and great coverage of cellular depletion at ACR. Take care.
All right, thanks.
Thanks, Jack.